Hello, my name is Rafael Martinez, and this is my poster audition video for the 2025 UCI Undergraduate Research Symposium. In this video, I will be discussing what inspired me to pursue my research project, the potential impact my project has had on the world at large, a general overview of my research project, and my overall experience conducting research in the McKickian lab over the past school year. As a first-year transfer student, my initial interest in conducting research at UCI stems from a biochemistry course I took, where I had to read research papers that were heavily focused on molecular biology and biochemistry. These papers I read drove me to look for pathology and biochemistry labs at UCI during the summer before my matriculation, which is how I discovered the McKickian lab. What motivated me to pursue my UROP project in particular was its focus on understanding subcellular interactions between proteins and their environment, which was something I found deeply interesting about the research papers that I had read. For me, pursuing my research project was just an opportunity for me to try to make my own discoveries about subcellular mechanisms while also improving my skills as a researcher in a wet lab. And over the past school year, I've learned much about many of the techniques used for cell culture and glycobiology research. So the McKickian lab is primarily focused on understanding the behavior of glycoproteins within immune cells, which are proteins with attached carbohydrates called glycans. If you treat cells with a compound called kifinensine, or KIF for short, the N-glycosylation pathway, which is one such pathway by which these glycans are attached to glycoproteins, is inhibited. As a result, many glycoproteins will have truncated N-glycans, and previous efforts in the McKickian lab have demonstrated that there are many glycoproteins that, in KIF-treated cells, are either significantly upregulated or downregulated at the cell surface relative to untreated cells. My research was focused on how KIF treatment downregulated most ZIP transporters at the cell surface, which are transporters that bring zinc and in some cases bicarbonate into the cell. Learning about the behavior of metal ion transporters like ZIP transporters has relevant clinical applications, as the dysregulation of intracellular pools of metal ions like zinc is a common marker in the progression of many types of cancer and neurodegenerative disorders. By conducting a number of experiments and functional assays using flow cytometry and Western blots, I was able to both confirm that KIF treatment downregulates ZIP transporter levels at the cell surface and observe what consequences this downregulation had on intracellular conditions, including pH, intracellular zinc concentrations, and the endocytosis rates of these ZIP transporters. The discoveries I made through these experiments has also laid the foundation for future research efforts directed at understanding how ZIP transporters with truncated glycans behave differently from normal ZIP transporters. One set of experiments I hope to conduct in the near future aims to identify precisely what mechanism or mechanisms of endocytosis are responsible for the turnover of ZIP transporters at the cell surface, and how truncating the glycans on these ZIP transporters with KIF affects their rate of endocytosis. Thank you for watching.